Welcome to People Behind the Science. I'm Dr. Marie McNeely, and I am thrilled to have you joining me today for episode 705 with our guest, Dr. Mary Jo Ondrechen. Listeners, as you know, through our podcast, we are working to introduce the world to scientists, science careers, and some exciting science. And if you want to meet even more amazing researchers, you can visit us at peoplebehindthescience.com or get connected with us on Facebook or Twitter at PBT Science. And today, Mary Jo has some excellent stories to share. So get ready to meet another one of our wonderful people behind the science after this short sponsor message.
For 45 years, Lampire Biological Laboratories has been a key player in the life science industry. We are currently looking to add new talent to our rapidly growing team. For more information, email hr@lampire.com or call 215-795-2838. Come change the world with us.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Listeners, today I am thrilled to be speaking with our guest scientist, Dr. Mary Jo Ondrechen. So Mary Jo, welcome to our show today. How are you?
Thank you, Marie. I am just fine today.
Well, Mary Jo, we are so happy to have you with us today. And I'd like to start by telling our listeners a little bit about you and how you got to where you are today. So listeners, Mary Jo is professor of chemistry and chemical biology at Northeastern University. She earned her bachelor's degree in chemistry from Reed College, and she was awarded her PhD in chemistry and chemical physics from Northwestern University. Afterwards, she completed a postdoctoral fellowship at the University of Chicago and subsequently a NATO Postdoctoral Fellowship at Tel-Aviv University in Israel. Mary Jo joined the faculty at Northeastern University in 1980, and she's been awarded the Outstanding Native American Student Mentor Award in 2018 from the Society for Advancement of Chicanos, Hispanics, and Native Americans in Science, or SACNAS. And she has been dedicated to advocacy and activism for underrepresented communities in science and society, as well as conservation and stewardship of the earth. So, Mary Jo, today we're excited to get to know you both as a scientist and researcher, as well as a person as a whole. So, can you start by telling us what you like to do when you're not doing science?
When I'm not doing science, well, I like to run and I like to grow vegetables in my garden and I like to cook. And I'm interested in herbs and spices and medicinal plants.
Oh, wonderful. So how's the garden going this year so far?
Well, this is Massachusetts, so tomatoes don't go outside yet.
That's fair. That's fair. Well, Mary Jo, it's wonderful to get some insight into your interests and passions outside of science. And I understand that you're also quite the activist and advocate for things like underrepresented communities in science and stewardship of the earth and environmental conservation. So can you talk a little bit more about these aspects of your life?
Yes, I've worked on advocacy for including the public and tribal nations in decisions involving cleanup of environmental problems, addressing environmental problems. The local communities, the local tribes should always be consulted. And I've also advocated for the use of currently available technologies, the newest technologies, to clean up environmental problems. I've been active in the Native American science community. I served as chair of the board of the American Indian Science and Engineering Society, ASIS, and I'm really proud of our young people. We have over 160 young Native Americans, Alaska Natives, and Native Hawaiians who are studying STEM fields and aspire to become STEM professors at universities, tribal colleges, or other colleges. And a couple dozen of them are already in faculty positions. So we really are increasing the number of Native American people in faculty positions in the sciences. And I think that serves to inspire Native students and other students that this is something that we can do and make a contribution to. So it's very important that science be done by people with diverse backgrounds and diverse perspectives. It makes our discoveries better.
Absolutely. I couldn't agree more. And what has it been like for you, Mary Jo, seeing some of these people that you maybe helped encourage to pursue science now in these faculty positions?
It's very heartening. It's wonderful. It's beautiful.
I could not agree more. And I think you are doing tremendous work on this front. And you're also doing remarkable things in the lab. And I'd love to talk about your research as well today. So how do you describe what you do in your lab to people who aren't in your field or maybe are outside of science altogether?
Well, Marie, I'm a chemist, but I do theory and computation. And we like to understand how molecules work. So I work on enzymes. Enzymes are nature's catalysts. They're proteins that catalyze reactions. I work on genomics, developing methods and theories to interpret genomic data. And then we are also the computational side of drug discovery. We help medicinal chemists develop new drugs and new treatments and new diagnostics.
Very cool. I look forward to diving into some of the details of your projects as we go through our conversation today. But I think in science, there are often days where you need a little extra motivation and inspiration. And I love sharing various quotes and sayings and pieces of inspiration for our listeners out there. So do you have a favorite quote or a saying or something that really inspires you, Mary Jo?
There's light above me, there's strength within me, and there's security beneath my feet. That's how I remind myself to keep going when the going gets tough. That was something I learned from elders.
Well, I think that is a great way to keep yourself sort of centered, grounded, and motivated in the work that you do. And I know as a scientist, there are a lot of role models and mentors and people that you look up to and get to work with over the course of your career. So are there particular people who really motivate, inspire, and maybe serve as role models for you?
Well, it started off with my late parents. Where I grew up, there were opportunities to go down bad paths, shall we say, like crime and drugs and addictions and so forth. And my parents thought that if they put us on good paths, got us interested in good paths, that we might stay off the bad paths. So they encouraged us to get interested in science. I learned a lot about astronomy from my father. He was not a scientist, but he loved astronomy, and he taught me a lot about the sky. And my mom used to take us to a museum on the weekends to learn about natural history, natural science. So they really encouraged me. Dagmar Ringa, who's on the faculty at Brandeis, was a very good mentor for me to get into biomedical problems. Around the turn of the millennium, I took a sabbatical in her lab.
Oh, very cool. Well, it sounds like you've had a wonderful set of mentors, starting with your parents, these people who really got you excited and interested in science. So can you maybe take us all the way back to the beginning and tell us a little bit more about these early scientific pursuits? You mentioned astronomy, kind of getting interested in natural history. What made you start thinking about science as a potential career path for you?
Well, back in the 50s, the U.S. was very upset about Sputnik. So there was a lot of encouragement in the wider culture, in the pop culture, to encourage kids to get into science. So I always thought that science was cool and science was interesting. And then I had a chemistry teacher in high school. Her name was Mrs. Prine. And Mrs. Prine really made chemistry fun. She was always doing experiments and having us do experiments, and she just got a big kick out of chemistry. She made it really fun. So I said, okay, well, maybe I'm going to do chemistry.
Very cool. And you pursued chemistry, of course, in college at Reed College. And then, of course, your PhD. How did you decide that you wanted to go on further in the academic path and get that PhD at Northwestern?
Well, I think that I wanted to be a professor, and I knew that to be a professor, you have to get a PhD. And not everyone encouraged me. It was not so common for girls to get into science back then. And some people were more encouraging than others, let's put it that way.
So what was that experience like for you then, transitioning from your undergraduate degree to now this kind of more professional PhD degree where you're getting more involved in research in the science?
Well, I got very good preparation at Reed College. I was in really good shape going into graduate school. So I felt ready. And back then, not every professor would take a woman in his research group. There were no women on the faculty at Northwestern at the time. But Mark Ratner was my PhD advisor, and he didn't care that I was a woman. There were other faculty at the time who did care, even though Title IX had passed at that point and it was illegal to discriminate. There were a couple of men who made it clear that they did not like having women in their group. That's all gone now. That was back in the 70s, and that doesn't happen anymore. But I was lucky to find an advisor who encouraged me and was very supportive of me.
That's wonderful. And then how did you decide on the next steps you would take in your career? I think this is often a point where students sort of stumble what they want to do after graduate school.
Well, to get a professor's job, I needed a little more research experience. So I took a postdoc at the University of Chicago, and then I took another postdoc with the NATO Fellowship at Tel-Aviv University in Israel. Tel-Aviv University had a very strong program in my field, and they still do.
I think having this opportunity to travel so early on in your career is remarkable. Was it difficult for you to transition from your whole career had been in the U.S. thus far to sort of picking up, packing up, and moving to Tel Aviv?
It was an adventure. I loved it. That was not my first time living abroad. My family lived in Japan for a couple years when I was a child. So I did kindergarten and first grade in Japan.
Very cool. So then can you tell us how you got from that point, wrapping up your postdoc there in Tel Aviv, to where you are today? What were some of these key moments that helped you find your way to Northeastern?
It was difficult back then. It's not so difficult anymore to interview for faculty jobs in the U.S. when you're postdocing overseas. But I flew back to the U.S. to take some interviews, and I was offered this job at Northeastern University. And Northeastern University has really been supportive of me, and the university has moved up in the rankings. The research mission has expanded exponentially in the time I've been here. So I feel lucky to have landed in an institution that's on the way up like that.
Absolutely. And you've been able to do some incredible work there at Northeastern University. So is there a particular project that you have going on in your lab right now that you are perhaps the most excited about at the moment, Mary Jo, that you want to tell us about in a little bit more detail?
Well, we're studying how enzymes work. And this work is supported by the National Science Foundation, the Molecular Cellular Biosciences Division. Now, enzymes, as I mentioned, are proteins that catalyze reactions, and they're really amazing. I mean, you're sitting here listening to this podcast, and maybe some people are in their cars or somewhere. And while you're listening, your enzymes are busy catalyzing reactions. You got hundreds and hundreds of them. They're busy catalyzing reactions. Now, if you or any chemist wanted to repeat some of those reactions in the laboratory, those reactions might require high temperature or caustic conditions, strong acid, strong base, strong oxidant. And yet your enzymes are making these chemical reactions happen at physiological temperature and neutral pH. So they're really amazing, and we want to understand how they do this, because understanding how these enzymatic processes work is important for understanding healthy and disease states in humans, to understand pathogens, for example. But also, suppose we could take the chemical industry, and instead of having thermal processes or conventionally catalyzed processes, we converted the chemical industry into enzyme catalyzed processes. Those processes would use substantially less energy, and they would also produce way fewer unwanted byproducts, because enzymes are very, very specific. They do their one reaction to produce their one product, and that's it. So if we could understand how to design enzymes to catalyze industrial reactions, we could save a whole bunch of energy and make the chemical industry far cleaner. Some of this is done now in the pharmaceutical industry, where reactions are catalyzed by enzymes, because the pharmaceutical industry typically requires a lot of steps in the lab to make. And some of these steps are catalyzed by enzymes. But we would like to take the whole chemical industry and convert it to enzyme catalyzed reactions. But for most industrial chemical reactions, you cannot find a natural enzyme that will do the job, so you have to engineer a new one. And right now, there are no best practices, no guiding principles for enzyme engineering. How do I make an enzyme that will catalyze an unnatural reaction? So we're trying to figure out all those pieces to be able to engineer enzymes in the future. So we're busy studying how enzymes work and looking at a few test cases.
Absolutely. I think this is a really exciting and really promising area of research, engineering new enzymes. And I love that you made us take a moment there to appreciate all of the hard work that our bodies and our own enzymes are doing, even when we feel like we're doing absolutely nothing.
Yes, your enzymes never quit.
Exactly. And I think our bodies are doing remarkable things, and trying to recreate these in the lab can be a challenge. So is there, I guess, a particular enzyme or process that you're focusing on with this research right now?
Well, we're working on one enzyme, a human enzyme called phosphoglucosideomerase, and it's involved in sugar metabolism. And we are running some calculations to explain experimental data about this enzyme, because we have experimental data that we published a few years back that shows that amino acids that are not really close, they're pretty far away from the point of reaction, are critical to catalyzing the reaction. They're not in direct contact with the reacting molecules, because when an enzyme catalyzes a reaction, the substrate molecule, the starting material, has to bind into the enzyme. Then it undergoes reaction, and then the product gets released. Proteins are made up of amino acids. So there's certain amino acids that make contact with that reacting molecule. And we know that those are probably very important for the reaction, but we showed that we can predict that amino acids not close, but maybe 2 or 3 layers away, are also important. We can predict which ones are important, and if you mutate them away, the rate of reaction falls. The enzyme is not as effective. So these distant amino acids are helping to guide the reaction, and it's not clear why. So we're running some calculations to explain exactly why this sort of action at a distance, as they say in physics, that these amino acids not close to the site of reaction are very important for guiding the reaction.
Well, this is fascinating. I know when you're pioneering a new area of science and trying to solve these mysteries, there are a lot of challenges. And Mary Jo, we love sharing the challenges, the struggles of science on our show, because I think these just aren't talked about enough. So do you have an example from your own research, from your own career, of a time where you really struggled or failed majorly? And can you tell us about how you worked through that difficult time?
Well, I think everybody who does research knows what failure is. Mm-hmm. Everybody who writes grant proposals knows what failure is. But you have to persist. And one of the best pieces of advice I got as a young faculty member, I was 27 when I joined the faculty, I was told that you just have to persist. You just have to have dogged persistence. And that turned out to be a very good piece of advice. If a proposal gets denied, you make it better and submit it again. I know a fellow at another university who got a multi-million-dollar grant on his 7th try. So that's persistence. But yeah, a lot of times something doesn't work, and you have to think about why and try to find a workaround. I mean, basically, that's one of the things that you have to do the most, is to figure out workarounds.
Definitely. I think that is a huge part of science. Do you have maybe a specific example of a grant that you were really excited about that didn't ultimately pan out, or maybe just one of these everyday research challenges or struggles that you run into in the lab?
Well, once we get the grant, we're able to do the work if we can do something with it. And it might not be the exact same thing we proposed to do, because the science has taken us in a new direction. But our funded projects have worked out very nicely. I think that we're theorists. We do computation and theory, but most of our work is in collaboration with experimentalists. And for us, it's a little easier to make a calculation work or modify a theory and make it work than it is for somebody who's trying to get a reaction to go or trying to purify a protein, and the protein just doesn't want to purify. So yeah, we've had joint projects where the experimentalists have had trouble purifying proteins. Some proteins just don't want to purify, just don't want to come together. So these things happen, yeah. There are other times we're working on something and we found something really great and wonderful, and before we get the paper published, someone else publishes it.
Yep.
That happened to me one time.
Oh, no.
So you have to dust yourself off when these things happen and keep going, because a lot of times things do work out very well. And we've had a lot of successes and we've had a lot of fun. So even though there are setbacks and disappointments and challenges, most of the time things go pretty well and they're fun. And you have to learn to see the challenges and the difficulties as a fun puzzle.
Absolutely. I think there's that mindset facet of it for sure. And I think if you get so hung up on these failures, these challenges, you miss some of the opportunities for exciting successes. And I do want to make sure we get a chance to talk about some of your successes. Mary Jo, do you have a favorite success story or example from your research that you'd like to share?
Well, we discovered a way to predict. If I have the structure of any protein molecule, I could predict which amino acids in that protein molecule are biochemically active. Are they active in catalyzing reactions? Are they active in binding another molecule? We discovered a way to predict which amino acids in a protein structure are active biochemically. And that's turned out to inform lots and lots of other questions. Like with structural genomics, with the genome sequencing, thousands of species have now had their genome sequenced. And from genome sequencing, the structures of many previously unknown proteins, there's three-dimensional structures have been reported. So there are like 15,000 structures of newly discovered proteins with unknown or uncertain biochemical function. So some of our work we've used to figure out what is the function of these protein structures of unknown function. We've used the work on SARS-CoV-2, the virus that was responsible for COVID-19, looking at the target proteins from the virus and figuring out where can molecules bind. Now, some of that is obvious, like this type of virus has a protease, and you could treat viral diseases sometimes with protease inhibitors, like HIV. People with HIV/AIDS take protease inhibitors very often. And there are protease inhibitors for viruses related to SARS-CoV-2. So it was pretty clear where the protease active site is in the molecule, but we also predicted other sites on the protein that are likely active in some kind of biochemistry, probably recognizing parts of the protein that the protease cleaves. So our ability to predict biochemically active amino acids and proteins has taken us down a bunch of new and exciting paths, including responding to the COVID-19 pandemic.
Absolutely. And I guess these prediction methods, are they pretty universal? Like once you have the methods established, can you apply them to a lot of different things? Or is it going back to the drawing board and solving a new puzzle each time?
No, we don't go back to the drawing board. It works for every protein if you know the three-dimensional structure.
Oh, wow.
Now, the crystallographers and the NMR people and the cryo-electron microscope people, the cryo-EM people, are very good at determining protein structures. There are some proteins that are very hard to determine the three-dimensional structure, but now there's a machine learning program called AlphaFold 2 that is very good at predicting the three-dimensional structure of proteins if you have the sequence. So that opens the door to millions of proteins that we can now characterize with their active sites. So yes, as long as we have the three-dimensional structure, I will tell you which amino acids are biochemically active, and we can now do it even for proteins that nobody's determined the structure of, because AlphaFold is pretty reliable in predicting what the structure of a protein is.
That is absolutely remarkable, Mary Jo. So I have to ask, how long did it take to get to this point on this process?
Predicting which amino acids are active started in 2001. And then in the following years, we refined the method to make it more accurate. We invented a new machine learning method to make the predictions even better. And once we invented that machine learning method, then we started applying it to all kinds of other problems, like predicting the function of protein structures of unknown function, applying it to drug discovery. And now our big project, as I mentioned earlier, is understanding how enzymes work.
Absolutely. And I think when you have an ongoing project like this that you continue to optimize and apply to different things, it's sort of hard to identify when you celebrate. So did you have an aha moment or a point of celebration in this process where you're like, okay, we hit a major milestone. We can take a moment to say, yeah, good job.
We celebrate every time a grant gets funded. We celebrate every time a paper gets accepted. So we have lots of excuses to celebrate.
That is absolutely wonderful. And I completely support celebrating every small victory along the way. And I know one of the aspects of science we have not yet had a chance to chat about are some of the opportunities that you've had to travel. Mary Jo, I think this is a wonderful aspect of careers in science. And we mentioned you were able to study abroad during your postdoc in Tel Aviv, but do you have a favorite place that you've gone for science, whether it was for training, to work with collaborators, for a conference, etc.? What are some of your favorite science travel memories?
That's one of the great things about this job, that you get to go to lots of interesting places. And it's hard to designate a favorite. I took a sabbatical in Aarhus, Denmark. Denmark's a lovely country. And I spent a month in Zurich, Switzerland, working with people there at ETH. And in 2021, I was on sabbatical in Budapest, Hungary, on a Fulbright Fellowship. And that was a very nice experience. And for my U.S. PhD students, my American PhD students, at least before the pandemic, I'd like to give each of them an international experience, to send them to a conference overseas or to work with a lab abroad. And that kind of got ruined during the pandemic, but I'm going to try to reinstate that.
I think that is a wonderful opportunity and a really great idea to introduce these early career scientists to sort of a completely different research environment in a new country and broaden their horizons that way. And you mentioned this 2021 sabbatical in Budapest. What was that like for you?
Oh, well, every morning I would walk to work along the Danube, and it was just a lot of fun. My host, Juliana Ola, was wonderful. And I learned a lot of new things, and we're still working together. I was back in Budapest a few weeks ago to finish up a project that we had started. So, yeah, it was a wonderful experience. The people are very friendly. It was a good time.
That sounds amazing. It sounds like you've been there a couple times now. What is something that you think our listeners absolutely must see or do if they have a chance to visit Budapest?
Oh, boy. The music is out of this world. It's a great country for music. There are all kinds of excellent concerts, orchestras. Sometimes restaurants will have live musicians. Just an excellent place to hear great music. And the food is good, too.
It sounds like a fantastic place to visit. I'm glad you had a chance to go. And listeners, if you're able to go, definitely plan a concert or two and make sure to sample the local cuisine. And we've talked about some of these collaborators, these people you've been able to work with in science, as well as your trainees. And I think oftentimes the scientists themselves are part of what makes the career so great, because I know there's these media representations of scientists often as very serious and critical and these almost robot-like creatures working in the lab. And that is just not the case for so many of the scientists that I've met. So do you have a story or an example of your own, Mary Jo, of maybe a quirky tradition or a funny memory or just something that goes against this stereotype that people have that really shows the human side of science that you could share?
Okay, well, I was just at the American Chemical Society national meeting in Indianapolis.
Oh, wonderful.
And a lot of chemists love to drink beer. And there's this character called Professor Millennium, because 6.02 times 10 to the 23rd molecules of something is called a mole of that thing. So this character who in a mole costume is called Professor Millennium. So I and others were photographed with Professor Millennium, and I posted a picture of myself and titled it "Two Chemistry Professors." People might not think that, but chemists are a very lively, fun bunch. If you go to one of our conferences, you'll see.
I absolutely love it. So this character, this Professor Millennium, is it like the animal mole?
Yes.
Oh, very cool. Well, I'm glad you were able to snap a photo to commemorate that moment. And it sounds like you have a wonderful time at these different meetings. And I know at these meetings, you get to have conversations with a lot of different people in your field, often from all over the world. And you can start brainstorming and thinking about these big questions in science that you're all trying to answer. So, Mary Jo, if we took away the barriers that normally hold you back, things like funding, staff, technology, time, feasibility, whatever it might be, do you have a particular question in science that you would want to answer most?
Oh, yeah. Interpret the genome and understand all the enzymes out there, all the proteins, everything. By the way, my Twitter handle is MJ Chemist. And if you look at my Twitter page, you'll see a picture of me with Professor Millennium.
Oh, we are going to have to check this out, listeners. And Mary Jo, I love this kind of big picture question. You're thinking about just sort of interpreting the genome and being able to sort of understand all of the wide variety of different enzymes and proteins that are out there in the world. What would you say is maybe the biggest barrier that keeps you from making substantial progress in these areas?
I think we're making good progress. There are just lots and lots and lots of them out there. And there's just not enough resources worldwide to characterize everything. But we're making good progress. I think it's an exciting time to be a scientist because there's just so many new things are happening now. The paradigm, the way that cancer is approached, for example, is getting away from tissue type-based therapies to genotype-based therapies. And that in the past, a drug might be tested on, say, 100 people with a serious illness, and the drug fails for 90 of them, but 10 of them get better. And they're clearly helped by the drug, but, 90% of the people were not helped, so therefore the drug is a failure and thrown out. But now it's possible to ask the question, what was different about those 10 people that were helped? And it's all in the genomics. It's all in the genes. So with gene sequencing, there are ways to personalize treatment, and figuring out all those details is very challenging, but very exciting.
Definitely. I think moving ever increasingly towards this personalized precision medicine ideal is an amazingly exciting part of being in science, and this biomedical science in particular. And there's just so much more to discover. Yeah.
Yeah, I agree.
Well, thank you for sharing your perspectives. I would love to end our conversation, Mary Jo, with some advice. So when you think back on your career, is there a particular piece of advice that someone maybe gave you at a critical point that really helped you that you can pass on to our listeners today?
Believe in yourself and keep going. I think that everyone, if you're interested in science, go for it.
Oh, I think that is amazing. Listeners, definitely take this advice to heart. Mary Jo, do you have any other last piece of advice or inspiration that you would like to leave everyone with at the end of our conversation today?
Well, thank you, Marie, for the conversation, and thanks to everyone listening out there. I hope that you learned something, and I hope that you'll be thinking about science.
Absolutely. And if our listeners want to continue thinking about your science and they want to learn more about what you do, what is the best way for them to do so?
You could Google my name, or you can look for my profile on the Northeastern University website, College of Science, Northeastern University, and my name, Mary Jo Ondrechen. You could also follow me on Twitter at MJChemist.
Well, listeners, definitely check out that website and get connected with Mary Jo on Twitter. And Mary Jo, thank you again so much for your time today.
Nyawa, thank you.
Thank you. It's been a pleasure. Listeners, great to have you here with us as well. We hope you'll join us again next time for another episode of People Behind the Science.